the ribosomes. So what we'll find as that messenger RNA strand leaves the nucleus, a large subunit and a small subunit will see or note or pick up the chemicals, whatever the case might be, of that messenger RNA strand and attach to it. The ribosomes are pretty cool. They have little docking stations inside them. A site, P site, but anyway, what this does, every strand will have a start codon, basically saying start the translation. Every strand will have a stop codon, stop the translation. But anyway, as the first 3 bases move into that first spot, I've got 1, 2, 3. This ribosome sending off signals, okay, tells a tRNA to meet it. The tRNA needs to be carrying complementary bases. Is everybody with me so far? The tRNA, well, in my ribosome, I got a spot for 3, so the tRNA carries 3. It comes in, docks, and matches up. Now, there's a second, but it's actually a couple, but anyway, in the next spot of the ribosome, I've got the same thing occurring, and guess what it brings with it? The tRNA brings with it an amino acid. So, as the messenger RNA strand moves through, a tRNA docks, matches up to it, brings its amino acid, the second one's doing the same thing, a bond gets created, this one moves out of the way, next one shifts in, next one drops in, brings its amino acid, and it continues to do this for the entire strand of the messenger RNA. For every 3 bases that exist, the tRNA that matches up to it brings its amino acid, bonds get created, and I now have my primary structure of my protein. So, if that's taking place, and I begin to put together my primary structure from every 3 bases, I am now getting what's going to hopefully become the structure of my protein. That ribosome will dock onto endoplasmic reticulum, continue forming the strand, and continue the process moving into Golgi apparatus. We'll either have something stay in the cell, or we'll have something that'll leave the cell. Those 2 processes, transcription, translation, are going to begin the making of our proteins.
So just out of curiosity, like, what makes the RNA and like the tRNA attracted to each other, and like the ribosomes, like chemical attraction?
Chemical signals.
Okay.
Yes. What will happen is, depending upon the cell of your body, okay, and what the product of that cell should be, that DNA that's in there, all 46 molecules do not need to be active. Only the ones that carry the genes to make the proteins need to be active. And everything is based on chemical signals. So, this is our product of making a protein. Now remember, proteins, everything is done by proteins, meaning the only thing that a gene can code for is a protein. But that mechanism that's in place has made you what you are. It has made it, whether you've got attached earlobes or unattached, whether you've got one eyebrow or two, okay? Whether you've got a widow's peak or not, okay? Whether the second toe is bigger than the big toe, all right? It has made everything that you are, all right? Every bit of it was based on chemical signals. So everything that you see, your phenotype, okay, in the mirror, all right, everything you see is based on this. Everything that's on the inside, your genotype, is based on this. Okay, it's making you who you are, carrying out all the processes of your body. Now, do the genes between me and you have to differ a lot? No, okay? We have very minor, very small amounts that will differ, all right? Yes, the genetics of mom and dad play into that, okay? But so far as the inside workings, we all have to produce proteins. We've got to produce insulin. We've got to produce stuff to break down the sugars, break down the fat. Do you see where I'm going? Okay? So, this would be expressing the genes. The genes are carrying the information. Now, in that process of transcription, where that DNA has a section of it open and that gene is available, that is where RNA will come in, match up to the bases, make my messenger RNA strand, and be able to leave the nucleus. This is the only time that the DNA, this is the only time information about the DNA leaves. Other than that, DNA stays locked up in the nucleus, safe and sound. Because DNA codes for everything. Anything that damages the DNA damages the proteins, and therefore whatever is supposed to happen in the body. When we have the process of transcription occur, have you ever wondered, how many of y'all maybe watch the crime scene shows, like CSI, Law and Order, okay, all this sort of stuff? Have you ever wondered how they match up the DNA? Have you ever wondered what makes that happen? Do you know what makes that happen? Okay, and pretty much now, it's hard to carry out a crime and not leave DNA behind. It doesn't matter how much you kind of like wrap everything up. But anyway, when you look at DNA, when you look at that makeup of it, we have what's called non-coding DNA. Coding DNA is what's coding for proteins and so forth in your body. But throughout the rest of that DNA strand, you've got sections of it that are non-coding. They don't code for anything. But that is special only to you. It's your genetic blueprint. My non-coding DNA would not be the same sequence as yours. But it's not coding for anything. Now, People who are truly identical. I like identical twins, but then you've got to be truly identical, split from the same egg. Okay? Will it be the same? Other than that, it's not. Okay? And this is how they match up stuff from crime scenes. So, in making a protein that we need, the cell has to cut out that nonsense material. Basically, that non-coding DNA, we need to get rid of it. And it will. So, we have to do some processing of that messenger RNA strand and keep only what we need so that it will code for the protein that needs to be made. So this would be post-transcriptional processing. So it would happen after transcription, before translation. And that's what that leftover portion that's kept, okay, is what will move forward and make the proteins. Like I said earlier, for translation to occur, we need the ribosomes. We are going to need the tRNA. We're going to need our 3 bases. We're going to need that to go into a messenger RNA to go into the ribosome. At the ribosome, have transfer RNA come in, bring amino acids, and string them together. We've got to make the bonds between the amino acids to be able to get the proteins. Take a break. Be back at 3:30.